Hello everyone. Did you have the good meals lunch? And my name is Jung-Hoon Moon. I'm in charge of the APRP session 1. And the APRP meaning is ASEAN Pacific Research Platform. The research platform comes from this 10 years ago, based on this science DMG technology. So currently the European and America and the ASEAN areas, they have their big data superhighway and combine the research, the computing resource as the research platform. So today we want to talking about is several the big data issue, the focusing on the scientific area, and the network issue, and all the others are that comes from the application issues. The many people want to this running their jobs as a code level or some delivery or the archiving and the sharing big data or something like that. But the computer resource and the IT infrastructure is not enough to support all our users. But the APRP is aimed to the support is based on the big data superhighway. It means that is NN and the 10 or each countries and the research education network. So the session 1 is consists of the 5 presenters, and the first speaker is Asif Raja from the Fermilab in the US. The contribution is managing the high energy physics is communicated with the US to the European CERN research laboratory. So Asif Raja, are you ready?
Yeah, can you hear me?
Yes, I can hear you well. And whenever you're ready and to start.
Yeah, I'm ready. So let me share my presentation. Okay. Okay, can you see my presentation?
Yeah.
Okay.
Yes, sir.
Okay. Thank you, Amun.
Yeah.
My name is Asif Raza, and I'm working here at Fermilab as a network and software R&D researcher, and particularly for US CMS. And today I'm going to present about the exascale data challenges and the networking R&D efforts here at the Fermilab related to the high-LUMI LHC. So I'll discuss something about the high-LUMI LHC, CMS, and the networking efforts here. First, these are the contents of my presentation. So today I think everybody know about the CMS and US CMS, but just for an overview, I am going to, start from here, and then I'll discuss about the high-LUMI LHC and the data challenges. And finally, the main important topic that I'm going to discuss today is related to our networking R&D efforts here at the Fermilab. And in that, I'm going to discuss about the Sense Rusio testbed at Fermilab, the Noted testbed at Fermilab, and our AI-based traffic classification using the ESNet high-touch services. So, as I said, most of the people know about the CMS experiment at the CERN. So it's basically a high energy physics and general purpose experiment, which is designed to precision study of the proton to proton collisions at, the CERN. And more than, 200 institutes are participating in CMS experiment, and around 57 countries, are participating in this experiment. And from all of the world, more than 3,300 physicists are, participating in this experiment. So CMS, it consists of the 9 tier 1 sites of the CERN and more than 50 tier 2 sites around the world. Basically, the compute needs of the CMS are mainly covered using the WLCG grid resources, which is highly distributed and global. It's a global collaboration of, you can say that more than 170 computing centers around the world and aggregating 1 million CPU cores and more than 1 exabyte of storage. So this is all about the CMS worldwide. If I talk about the US CMS, Fermi is one of the largest tier 1 center for CMS experiment, which is holding more than 40% of the data in US, the entire CMS. And along with the tier 1, we also have the 7 tier 2 sites as well. And basically, the purpose of the US CMS is to enable the physicists to take the advantage and the leadership role in the CMS physics. If we talk about the current era of the CMS and LHC era, which we call it the current era is the LHC era. But if we look into the future, that will be, the HL LHC era, which is called the high luminosity LHC era, which will start from the 2029. I'll discuss more about that thing. So if we look into the high lumi LHC era, so there are lots of research and development efforts are ongoing around the world, including in our US CMS. So we are trying to work on some R&D efforts for US CMS to enable the HL-LHC era in future. So what is HL-LHC? Basically, this is the next phase of the science harvest at CERN, which will start from the 2029. And it basically, if I talk about particularly only for the CMS, it will generate huge amount of data, which can be, that 20 times or 15 times larger than the current. You can see in this table that how much data we are currently generating per year. And in the future, in HL-LHC era, it will be 600, petabytes of data. So to consider these challenges, to fulfill the requirement of the HL-LHC era, community build, they establish some data challenges events, biannually, you can say, means after 2 years. So the purpose of these challenges is to demonstrate that if we have the huge amount of data and we want to transfer storage to storage, then how our infrastructure is capable of, transferring that huge amount of data to reach, till the 2029 to accommodate the HL-LHC era requirements. So these are some targets. Here you can see this is the Fermi Labs US FNAL, and we have some target also. The total capacity target is 800 Gbps links and network through which we can transfer the data. And in 2021, we had the target. It was a DC 2021 event happened, and we achieved that target, which is basically 80 Gbps link, and we have to fill that bandwidth by transferring the storage to storage. And currently, the next event of the DC24 is going to be happen in this February, and it will start from the 13th of the February. And our target is 200 Gbps link. So that's basically what the target we have for DC24 to achieve the HL-LHC era in future gradually. So these are some efforts that R&D efforts for HL-LHC era in different areas, including the networking. So you can see the data management softwares, because when we are talking about the HL-LHC era, we are not only talking about the networks, we are talking about all the software services and storage technologies, networking technologies, what will be the challenges for the storage, what will be the challenges for data transfer technologies, data management softwares, and networking as well. So my focus is mainly on the networking R&D efforts here at Fermi Lab. So I'm going to discuss about that thing in depth. So if we talk about the future needs and requirements, networking requirements of HL-LHC era, as I discussed, that we have to achieve at least 800 at Fermilab only, we have to achieve the 800 Gbps link or more than that, which is maybe 1.2 terabytes per second. So, but the question is that when we are just increasing the link capacity, is it enough to just increase the link capacity? Obviously not, but we have to utilize that link in very efficient way. So considering that point, we are working on 3 different projects, basically through which we can utilize our networking links very efficiently in future. These are the 3, softwares. These are the 3 test beds, I can say, that on which we are working on, and we have the test bed at Fermilab. I'll discuss about one by one. One is the Sense-Rusio architecture, another is the Noted architecture, and third project is basically the AI-based traffic classification. So let me discuss first about the Sense-Rusio. Basically, Sense-Rusio architecture is an end-to-end service which basically dynamically provision the end-to-end path, which is a QoS guaranteed path, which is a priority-based path. Whenever we are transferring the huge amount of data from one side to another side, I'm considering the CMS data here because we are dealing with the CMS data right now. So then we need, QoS guaranteed path, we need, priority path, we need, the traffic engineering, and we want to, know about the deterministic paths between 2 endpoints. So we are using this Sense architecture here at Fermilab, and we establish a test bed of the Sense architecture here at the Fermilab. How it is basically working, Sense orchestrator is basically communicating with the Rusio and FTS, and then it identify the requirement of data transfer, and it create the end-to-end path by using the network resource manager of Sense and the network side manager of the the Sense. So it creates the end-to-end QoS guaranteed and priority path whenever we require the large amount of data transfer from one end to another. And so in this way, we can efficiently utilize the our bandwidth that we have. So this is the test bed that we have at Fermilab. You can see this is our production routers, and this is our test bed router, and these are our servers. I'm just illustrating here in the the boxes, and this is our similar like the production environment we have at Fermilab in our tier 1 center. We have the Dcache storage and X2D servers. So all those things we are just duplicating here at our test bed, and we are demonstrating that how we can create the path using the Sense the architecture. And basically this is the collaborative project leading by the ESNet and the Lawrence Berkeley Lab and Fermilab, UCSD, Caltech, University of Nebraska. These are our tier 2 sites. So this is overall our US CMS test bed. This is only our Fermi's lab. We established the Fermi's test bed here inside the Fermi. And here you can see this is the overall test bed, which is connected via the ESNet and all the sites, the Caltech, the UCSD, Vanderbilt, Fermilab, and the University of Nebraska. So by using the Sense-Rusio architecture, we can create the end-to-end path among these, all the sites, and we can transfer the data whenever we require. We also demonstrated this in SC23, and you can see how we can, you can see here the results, how we can, prioritize the data, and we can achieve the, priority-based data transmission from one end to another end. Another, the test bed that we have is the Noted test bed, which is also similar like the Sense, but it is, both are complementing each other. Noted is basically leading by the Sun. So Sun is developed this Noted architecture. It's basically the network optimized transfer of experimental data. Whenever it will monitor the bandwidth and it will monitor the link, and whenever link is going to be congested, it will shifted the path from one path to another path. So that's basically the main concept of the Noted. And we also have, along with the Sense, we also have some Noted servers here deployed in our test bed, which is only dedicated for the noted testbed here at Fermilab. And we are also collaborating with the CERN and the ESNet here as well for this, because CERN is leading in this project and we are supporting them to deploy the noted testbed here. How noted is basically working here at Fermilab. Let's suppose this is the default path we have from Fermi to CERN for any data transfer. Let's suppose we initiated the data transfer and it's going via the LHC1 or LHC-OPN path. And when the path is going to be congested, it is going to be monitored by the FTS and the sense. Noted is going to be monitored the FTS queues. And when FTS queues are going to be congested, it will redirect the path with the alternate path, which is created using the sense or any other path as well. So in this way, basically the noted is working. And this is how the noted we demonstrated in SC23 as well. The third, the project that we are working on is AI-based traffic classification using the ESNet high-touch services. Basically, ESNet high-touch services is a part of ESNet 6, which is providing the precision network telemetry services to the sites where the ESNet is providing the services, network services. They also provide this telemetry service to sites. The purpose of this telemetry service is mainly two things. The flow deciliation means summarize the large flow in a small flow, and the network microscopic services means we can use those small flows and we can analyze further as we required. Here you can see in our Fermilab, there are two, the high-touch servers are deployed at our uplinks by the ESNet, and we will use these and we will, summarize the flows by using high touch, and we will analyze further with two different techniques. We are going to implement here the two different techniques. One is the deep analysis technique, and other is basically the AI based techniques, which is the machine learning and deep learning based techniques through which we can train the model. The purpose of this, the AI based traffic classification is because within the Fermilab we have very different sort of traffic in and out, and particularly we are focusing on CMS traffic, that we can identify, okay, this is a science traffic, this is a non-science traffic, this is a CMS traffic, and this is a DUNE traffic. And at the protocol level, at the application level, at the data and stream level, we can identify what type of traffic is going in and going out within the Fermilab. So therefore, currently, we just started this project with the ESNet, and we are working on this and hope so we will have some results in near future. So this is the third project that we are working here at the Fermilab. So this is all about for my presentation, these three, the test beds we have right now at Fermilab, on which I'm currently working on and working in the collaboration with other like the ESNet, CERN, and others as well. So mainly working in the networking R&D for the US CMS. So thank you. And if you have any question, you are always welcome.
Yes, thank you. Thank you, Asif. And does anybody question for the presentation? I want to ask something is, I just, the previous page, I saw this Xilinx FPGA card.
Yeah.
Yeah. How many do you have with the Xilinx FPGA card?
This is the high-touch server is the server for the, it's a part of the ESNet precision telemetry services. It is not our part. We are just using this server as a service and get the data at our end and classify that data. Basically, this is the one, whatever I know, I can explain you. The FPGA is the one FPGA we are using here. What we are doing, whatever the traffic is coming in and out from this router, it is going to be mirrored and goes to this FPGA. What FPGA is basically doing, it's monitoring the flows and just not complete flow. Let's suppose we are transferring a data which is more than 50 GB data from one location to another location. So we cannot store the whole entire flow packets. So what FPGAs do, it will monitor the real time, the flows, and just based on the 5 tuples, it will conclude summarize the information of the flows, and then store in the database. And then we use those flows at Fermilab, and we will classify that what type of traffic it is. So the purpose of the FPGA is to fastly process let's suppose we have the 400 Gbps link here at the Fermilab, and you can imagine with the normal NICs, we cannot process that much amount of the traffic on the 400 Gbps link. So therefore, they are using the FPGAs here.
Okay, thanks. I think this Xilinx FPGA's configuration is really hard.
It's
Anyway,
not my headache. Yeah.
yes, I will ask again to how to the configuration. Thank you for Asif. Yes. I want to introduce his next presenter. Is Asif Khan. Is from Malaysia, Feradana University. Asif Khan, are you there? Asif Khan?
Yep. I'm here.
And he engaged in the bioinformatics, and that's why he need this HPC like the computing environment. And are you ready? And go start.
Yeah, I'm ready. Okay, so let me share my screen. Zoom. Okay, can you see the screen?
Yes, it's where.
No.
Okay, good.
Okay. All right. Thank you, Moon, for the invitation to be presenting here. Hi, everyone. This will be a bit of a gear shift compared to the other Asif. I will be mostly talking about the biological aspects. And why is this relevant? This is more of a case study just to showcase to you how the computational power can be utilized to solve biological problems and really benefit, community at large. So just to give you an idea, and I'm sure there are many other examples, but in biology, I represent biology in this sense, and we have a big need for compute power, and many of us at the ground level don't really have access to this. And my coming to this APAN meeting as part of APRP, just to advocate and just to remind those of you who are having access to compute power that we need to find ways to bring this access to the masses, to the community who really need it. One big example, I had a master's students who, because of lack of compute power, had to do a project for, based on theoretical calculations, would have taken 7 months to finish. And that's just not acceptable in this time and age. And because we didn't have access, we tried to optimize the algorithm. We managed to bring it down to 2 months. With further, we managed to bring it down to, I think, 17 days eventually. With some access to compute, we were able to reduce it. Of course, we should always optimize our algorithms. We don't want to, feed compute with all kinds of non-optimized ones. But I think the two can meet somewhere at a midpoint. We optimize at the same time. If we have access to big compute, we will be able to do wonders. And today I'm going to talk about one of the other areas where I feel we had a similar limitation in terms of compute. All right, so the topic that I want to talk about is charting the viral human chorion. So the word chorion might be of interest, but viruses and human, of course, we are very familiar with that. By the way, just a correction, I'm not from Malaysia, Perdana. I moved recently. It's actually almost a year next month. So I'm right now with University of Doha for Science and Technology in Qatar, and happy to be part of today's presentation. So viruses, very quickly, we all know what they are. We just came out of COVID, and there's still, COVID scare in some parts of the world. So we are not completely out. But viruses are there, and they are actually the most abundant microbial agent in nature. Right? We don't want to go into the details of that. I think we know enough. And of course, we also know how lethal they can be. These are some of the most lethal viruses that have been documented that we are aware of in different parts of the world. So Ebola is very common in Africa. We have Marburg viruses, Zika viruses were a scare in South America and other parts of the world. Lassa fever virus, Crimean Congo, Rift Valley, Nipah viral. Some of these you may have never heard, but there are actually a lot more of these viruses that are really, really serious threat to human to our health. And MERS, we have heard of MERS. This is another coronavirus, and of course, who can forget SARS? All right, so this is just to remind us that there's a lot more going on besides SARS. And so what is this shariome that I'm interested in? So shariome is essentially a question we asked, sequences, that viruses are made of sequences, DNA sequences, and humans have DNA sequences. The simple question was, what proportion of viral sequences are exactly the same as human sequences? Do we have sequences in human that are identical to those in viruses? And of course, this has been reported before. We do know that viruses, when they infect humans, some parts of their genome becomes integrated into the human genome. HIV is a very famous example of that. All right. So in fact, a very interesting story is that they found these marsupials, like kangaroos, having parts of their genome with viruses that are found only in Africa today. So for example, Marburg viruses, they're found in Africa. And this was a big question, like why would marsupial kangaroos found in Australia? I think they are the only one, they are only there. Australia would have sequences from viruses in Africa. You could say this is a mistake, and of course they checked, and it was not a mistake. And of course, if you look at history and geography, we know that thousands of years ago, many years ago, the continents were connected, and there's drifting away, the plate tectonic movements. So at one time, Australia could have been, should have been, or must have been part of that one joint continent, and organisms or these organisms were moving around, and there was perhaps some infection integration of this happening thousands of years ago. So this also tells us a bit about what happened in the past. These sequences also represent footprints. They're like, footprints, fingerprint footprints left behind by events of the past. So it's a very interesting area because by knowing how much of it is in human, by knowing who are these coming from, we know the popular examples like HIV and others, but there might be other viruses that we are not even aware of. And by doing such a analysis, we will be able to identify who these viruses are and maybe do something about them, prepare them, maybe prepare ourselves to be able to tackle maybe potential future issues. So of course, these are real genuine integrations, but they could also be, just by chance happen to be the same as viruses. But that also has implications, because even though they are by chance in human being the same, they may share the same structure, they may share the same function. And we can also then learn something about function, something about structure, and see what that can teach us. So for the purpose of today's conversation, what is the definition of a shared sequence? Basically, it's something that is found between the two that is 100% identical. All right. Of course, we are not talking about the whole genome. We had to reduce it to a certain size. So we, decided to look at, as per the definition, can be partially or full length, but for the purpose of the analysis that we were going to do, we used monomer, basically minimum nine amino acids, or 9 amino acids were identical. We will call them shariome. Now, what are these shared sequences? What could they be doing? They actually are important for the life cycle of the virus. They can help viruses hijack the host machinery. Host here is the human machinery. They can help the virus avoid immune system of the host, and they can also alter the metabolic process of the human. So it's like something that benefits the virus by integrating. It's like you plant a spy, right? Within the houses of people that you're interested in, and they give you information, they help you, they facilitate, right? So our motivation was, take available data. There's a lot of sequence data for viruses available, and of course for human, that's publicly available for free, and it's available. Anyone can take it. You can access it. So the idea was to download this data and then analyze for shared sequences, and then try to understand what's going on. Right. So look at the structure, look at the function, and then look at what are the implications in terms of diagnostics, surveillance, treatment, vaccine, drugs, etc. Those are the implications of the study. So there's a lot of value in being able to do something like this. Now, so we wanted to do just viruses and human, But this is just an example because there's so much more that can be done. We can actually do this at a very specific level, viruses with a specific virus, let's say HIV, we can do it at HIV level, we can do it at the dengue level, we can even do it at a SARS level. There are thousands of species out there. You can choose to do it at any one of those levels. But what we did, we just did all viruses and all human sequences. But you can also replace human with something else, maybe, monkeys, you can even replace with other hosts, chicken, etc. Those that could be important for as a food for human. We could even look at those. So those are the potential applications of such a study. So the way we did it, we collected the data, we then processed the data, we tagged the information like what species is this sequence coming from, and then we broke those we removed the duplications, we removed duplicates which are quite common in these databases. The same sequence may be deposited by two different groups from different countries. So we remove duplicates, and then we do something called k-mer construction. We broke down the sequences into k-mer. The k-mer here, the length we chose was 9, because 9 was statistically significant enough to say if there is a match of 9, that should not be by chance. There is some meaning behind it. All right, so we chose 9 and we broke down all the sequences by 9, and we still know where they come from, so we have not lost that information. And then we remove duplicates of 9, because when you break long sequences into 9 mers, like 1 to 9, 2 to 10, 3 to 11, then obviously you may have duplicates coming from different sequences. So you remove the duplicates, you get something called unique k-mers, and then you have two sets, one from the human, one from the viruses. Then you just basically mash them, compare them to see for matches. A very simple analysis computationally. And what you will get will be the human viral shareome. And then you try to map back which species are they coming from, which proteins are they coming from, and etc. There's just so much that you can do in terms of understanding what the result means. I'm not going to go into the details of those analyses. So the resources we used, we built our own tool in order to be able to slice the sequences into whatever k-mers of choice. We used 9, but anyone can do different lengths of k-mers. And then we used a bunch of databases to download the data, but not just download, but also to characterize what do those sequences mean, what do they do in terms of maybe immune, maybe in terms of vaccine, in terms of drug design, etc., structure, function. And of course, we had other tools that facilitated all of this. Now, the restriction we had, downloading took time. So that was one computational challenge we had. It took about 2 months just to get all the data. All right, and this is the problem, that we are now liaising with NCBI, and I think there are much better approaches now in order to download, but still, it's not like you can just download within a day. This will still take at least a month. We have optimized, but still, the wait is still there, which is painful. Like 2 months are gone just downloading data. So if you have suggestions, if you have ways to overcome that, maybe we can talk about that. The other one was, of course, you have millions of sequences. You're talking about millions of sequences, and then when you break them into k-mers, they become billions of sequences, and then you have to remove duplicates. Then they become much smaller, right? And then you have to do the analysis. You have to compare. Imagine if you have 10 sequences for viruses, 10 k-mers for viruses, 10 k-mers for the humans. You have to do one by one comparison. You take the first one and you compare with all the 10, then take the second one, compare with all the 10. So you have to do that for comprehensive analysis, and maybe there's a way to optimize all that. So that is another area that is computationally very heavy, and it takes a lot of time to finish that. So the exciting part is to making sense, but we have to wait for quite some time in order to get there. And I would love to be able to do this for anything on the fly, but we are limited. We are by these computational requirements. So what did we get? I'm not going to bore you with the details, but I just want to maybe highlight, maybe just focus on this picture here. So, that's 100 million, k-mers from one side and 18 million k-mers from the other side. And what matched were 135,000 k-mers. So very small. Of course, we expected small, but now we have a precise number of what was the match. And this represents 0.13% of viruses. So from the viral sequence k-mers, this represents less than 1%. And on the other side, it's also less than 1%, but on the viruses, it's much smaller. But bottom line, less than 1% of the sequences are matching. But what is interesting is when you map these k-mers back to the actual protein, the full-length proteins, then it actually maps to 819,000 viral sequences and 164,000 human protein sequences. So these k-mers, small bits, they come from 164,000. That's a lot of different protein sequences that are actually having some elements of being shared with viruses. And what could they be doing? How are they helping viruses, or what their roles might be? That's the next big question that we tackled. And I don't want to go into the details of all that, but just to show you, like, we were able to trace back what is the family of viruses that are most common. So retroviridae, which is where HIV comes from, they were the most common. But we also had some other viruses that were quite dominant in terms of having these shared sequences. And of course, among retroviridae, among viral species, these are the species that were the most common. So HIV is number one. Then you have influenza. Surprisingly, influenza also has hepatitis B, virus, and so many other viruses that you see the names here. All right. Of course, for many of them, the distribution is very small, but nonetheless, they do have something. And then we looked at, the network analysis, and we found that some of these genes could be involved in cancer, even olfactory, like, your visual, your eye genes. And of course, there were many other areas that we found that were relevant, the kind of functions these sequences were playing. Right. So that's a whole different interesting area, but this is just to give you a taste of what we learned. And that's it. I don't want to go into the details of the biology, but I just wanted to highlight, that there's a huge potential. I would love to be able to do this, as quickly as possible to as many species. I think ideally we should do this for every single species, and we should come up with a map, with a catalog. Anyone should be able to go to a database. Okay, I want to know what are the shared sequences for this particular species, and what are they, where do they come from, which proteins, what do they do, what do we know. Imagine if we could have a database that detailed for every one of them. I think that would be a very valuable resource for medical practitioners to be able to then take that knowledge and maybe start early on preparing drugs, or preparing vaccines, or even diagnostics, so that we can do a better job in terms of tracking and managing diseases that are happening around the world, especially those viruses that are important for medical purposes. So in conclusion, it's a negligible fraction that is shared, less than 1%. However, they originated from a larger number of proteins that becomes then very important from medical point of view. So 18% for viruses, 13% for percentage-wise, they still seem small, but the absolute number is very high. All right, so their shared sequences are more pervasive than we expected. We perhaps expected them to be very small, but they're so much more. And of course, they have important functional, structural, and immunological relevance, which can help us to understand host-viral interaction. The data was important, as to make a point that more needs to be done, and these efforts would be valuable for surveillance, treatment, or even vaccine development. The methodology that we have defined, can be now automated to be able to reproduce such work, or to do such work for other viruses or other shareomes, whatever combo that you're interested in, one versus another. And it doesn't even have to be virus. It can be expanded to bacteria, fungi, you name it. Whatever you may be of interested, such a methodology can be applied. But again, it's the compute that's the challenge. And my take-home message for all of you in the audience would be, how you could help us biologists to be able to do this kind of work much quickly, much more, and be able to, maybe benefit the community through the computational resources that you have. So that we would have a much safer and much safer world, and less diseases, hopefully down the future. That's all from my side. Thank you very much.
Thank you, Asifu. And anybody question for this topic? Yeah. Yes, behind. Oh.
Hi, thank you, Asif. I'm a network engineer. I'm not a biologist. So what is the benefit of finding these overlaps, and so we understand what you'd be able to gain from finding the overlap in the share genome?
Right. I think just to highlight, first of all, by finding the overlaps, we will know what species are they coming from. So, for example, I was personally surprised that influenza has so much overlap. We didn't know that. Maybe you could expect some, but we see that the percentage or the count is rather high. So it kind of at least provides us, number one, knowledge of what we didn't expect and what actually is the reality, and that might shift importance. So now, I mean, HIV has received a lot of importance in terms of integration and how do we deal with that. But in terms of influenza, literature research that is looking into this, as far as I know, is very minimal or very limited. So this, number one, it gives us a perspective that we were not aware of, or an insight that we were not even thinking about. So and you could imagine there may be viruses that we just completely were unaware of. So they might then become important priority viruses that deserve further investigation. That's one. Then the other thing which I didn't really show, I didn't want to bore all of you, is like, what do these sequences mean in terms of function? What can they do in terms of structure? Just to give you one example, we have one virus called cytomegalovirus, and what it does is it actually codes a protein. The shape of that protein is exactly the same as one of our immune defense protein called HLA. So it's like a decoy. So now imagine you have a virus that's taking the same shape as a host protein, and now the host is going to be confused if someone is infected with that virus and there are proteins floating around that look like your host. And then what happens is if other proteins bind to that one, and then and it kind of does the job of the original one, but then it kind of stops halfway. So it doesn't do the full job, and that benefits the viruses. So it also helps us understand how viruses might be hijacking, might be taking advantage. So I think what I'm trying to say, the first thing should be we should have a roadmap. We should have a map of what exactly is the landscape of this Sharion, and then drill down to who they are and what could be what could they be doing in terms of structure, in terms of function, and that would hopefully better prepare us in terms of fighting these viruses, combating them, and avoiding them from detection, diagnostics, and drug development to inhibit them, or vaccine to prevent them. So that these are just some of the larger implications of the value that could be gained from such a work. I hope that gives you some idea.
Okay, that's great. Any other question? Okay, that's all. Thank you. Thank you again, Asipan.
Thank you. Thank you, Moon. Bye.
Later. Yes, I want to introduce this next speaker is Eve Pops. He came from the Singapore NSCC. He is the expert for this research platform areas. He's going to speaking about this the global research platform and those areas overall. Eve, are you there? Hi, Eve. So when you're ready and start your presentation. Okay.
Okay. Thank you very much, Jong-hoon. Happy to be here. Well, here in Montreal, it's minus 10 degrees Celsius, so it would be better in Bangkok, of course. But next time. So what I would like to share with you today are some thoughts on the research at the speed of thought and a road towards a global research platform. We have learned from Asif and Asif, the two application domains which really need a lot of compute, a lot of storage, and a lot of collaboration. And that's where that global research platform really comes from. So why do we have research platforms? It's essentially for big data and the sharing of science equipment. And the best example we know, of course, is the CERN in Geneva. Again, to have the CERN in Singapore, I think Singapore would be too small to have the whole ring. And so this is one domain, and they have been giving the example to all of us on how to build these research platforms. And again, they have had a kind of a closed environment, so it was a safe environment. So when we, of course, want to share these things, we need security, so we need DMZs and have the wide area networks connecting all these things used in a kind of a closed user groups. So basically, in the research platform, you will have partitions for the physics people, for the astronomy people, as we know, SKA with South Africa and Australia will also generate loads and loads of data when it comes online totally, a little bit simultaneously with our high luminosity LHC 2029. So towards the end of the decade, I think the data will be astronomical. The ITER, so the fusion research, they're also about to start generating loads and loads of data. And don't forget, and also the South America, all the telescopes in the Atacama Plateau, they will also generate huge amounts of data. And of course, our biology, as Asif said, our genomes are quite big. So if you want to have a lot of genomes and a lot of shareome, in fact, it's the first time I heard the notion of shareome. It's very interesting. I'll do some reading about it to be able to talk a little bit more intelligently about it. So this is another area, and also not to forget that we have global warming, the weather, etc., etc. So there are a lot of data and a lot of distributed data to be accessed. Now, the APRP, the Asia Pacific Platform, where Jong-Hoon Moon is a chair and Andrew Howard the co-chair, is an extension of the original PRP in California. And one of the elements I would like to stress is the Data Mover Challenge. The Data Mover Challenge is something we started in the context of the Supercomputing Asia conferences to go and try to find the optimum softwares to transmit. As again, it is quite important to have a relatively fast, in fact, very fast, depending on the application domains. If one would look at weather forecasting, for example, the validity of data is limited in time, so it has to be transmitted fast to the other side. If we want to look at in a very large baseline, there also cooperation is fast transmission. So the DMC challenge basically has the DTN, the data transfer nodes, at the end. So basically, the network of the Data Mover Challenge is in itself a research platform, and the research platform which should be used, and not only used for supercomputing every year, but used throughout the year. This latest edition of the DMC, so the Data Mover Challenge happens every 2 years now, and the results of the 2023-24 campaign will be announced at the Supercomputing Asia conference, which will happen next month in Australia. And of course, you're all very welcome to attend that conference. So I wanted to say that one of the interesting parts in the latest edition of the DMC was an initiative by our friends in Japan to try a satellite link and what the most efficient ways are to, to transfer data on the satellite links. As we know, cables break. There is also the geopolitical aspect where some countries threaten to cut cables, transmission cables of other countries. We've seen some strange things happening in the Baltic Sea, in South China Sea. So the satellite is very important, and going forward, I think it will be even more important. Now, Asia Connect has been a key catalyst for APRP, and we have to be thankful to Asia Connect. I think Jong-hoon will talk more about it, and Asif Khan has been participating also, and basically one of the major and most beautiful applications in Asia Connect has been the building a high bandwidth distributed HPC, which in fact is a model and which project has been concluded, but we should find ways to continue it because we need remote access to HPC. We need remote access to data. As Asif says, it takes sometimes a lot of time to go and access information on the other side. And obviously, with the compute and with the data, you need also fast processing. And that's where the shared HPC element comes in. Again, congratulations to Kisti, to Jong-hoon for what they did in the project. Again, this is a model for us to follow everywhere in Asia. Distributed computing with Pakistan, with Malaysia, with Australia, with Korea, of course, involved. Now, in Cyprus, this summer we had the fourth edition of the GRP. So the GRP is the global research platform. So the dream is to eventually connect all these discrete pieces of a research platform together and have one big one to be able to really collaborate on an international level. And in fact, I will not go into the detail here, but all the presentations are available on the GRP workshop. I've put the URL down here. I would recommend you to have a look. There are quite some interesting presentations in there. And next year, the GRP5 will be in Osaka, Japan, in August. As I said, the APRP comes from the US model. The first iteration was the Pacific Research Platform in California, running on the Scenic network. And they did an excellent job. And the NSF then awarded an extension to that first project, which was a TNRP towards a national research platform. And now the latest incarnation is NRP, which is an experimental research platform nationwide in the US, and which is quite interesting. And again, as of January this year, the NRP enables access to over 1,300 GPUs, 20,000 CPU cores, and 22 FPGAs. And connectivity at 40 to 100 gig between the pieces. And in fact, they call that, and I think it's the right name, the Cyber Infrastructure Commons. Commons in California, Commons. In fact, the same Commons you find in Singapore. As we know, the Korean research platform has the 25 national research centers connected. This is the way to go. CNIC and Calren is the home, as I mentioned. The next phase will use the Expanse, which is the SPC resource sharing developed at supercomputing in San Diego. And it's interesting. I would advise everybody to look what and how Expanse evolves. And then there's the Exceed part, of course. There's the Open Science Grid, which is also an interesting element because Frank Wertwein, who's the PI of NRP, also was one of the fathers of the Open Science Grid. So he is really in favor of open science. In fact, one beautiful project he had that was at the supercomputing, I think 2 or 3 years ago in the meantime, was to share GPUs on hundreds of computers to have a simulation of the ice cube neutrino detector at the South Pole. So this will be the NRP again. Cree is our component in California, plus, of course, the rest of the US helping. Larry Smart, who was PI, explains the Expanse. And in fact, the last paragraph, which I highlighted in red, is most important. I think you're going to see this type of cyber infrastructure become a permanent part of the research landscape. A completely distributed high-performance computing, networking, storage, and analysis facility. So this is true everywhere. Lessons learned: innovation, adoption, scaling, sustaining. Actively seek new collaborations, and that's where I think is the most important point. And the PRP, when the PRP started in 2015, one of the basic elements which led to PRP is to have the researchers who want to collaborate basically sign up and say, we will collaborate. We biologists need a research platform. We astronomers need a research platform. We weather people need a research platform, etc. Because too often platforms and networks are being built, then you go to the researchers and researchers have nothing to collaborate on, or they don't have money for projects to collaborate on. And from a personal experience, putting together the research network in Canada 25 years ago, we thought telemedicine will be the killer application. So we went through all the major research hospitals, and they were all very interested. And then they all said, how do we get your funding? They say, we don't fund your research, we fund the network. So basically, there is the need for the application domains to come up. Now, to conclude, the APRP suggested next steps: expand the footprint, use the DMC topology as a basic for a permanent GRP platform. In fact, that permanent GRP platform is also the equivalent is being discussed in SciNet. There are also some who would like to have the SciNet becoming more permanent. It would be less expensive to build, for one, and would also have then trials on a more regular basis. Excuse me. Another aspect is to set up an APAN directorate to identify funding resources. Again, without funding resources, we talk a lot, but nothing much happens. We have to give presentations and have visibility. And I would say APRP should plan a keynote at the next APAN. Another aspect, and there the US is really leading, is to standardize and document APRP implementation as our friends at the ESNet have been doing. The DTN configurations are, and the recommended configurations are quite well developed and well done. So, seek new collaborations with selected communities, we have said. Actively plan national science DMZs. And again, in Europe, we have not seen many science DMZs or many research platforms, I should say, coming. I hope they will be coming. And again, we have to evaluate our US experience with expands, with noted as if was mentioning in his Fermilab presentation. The P4 switches developed by the Europeans at Géant are quite interesting. So basically, in conclusion, we have to do something. And also one thing which I would stress is the international aspect, because when Asif Shah gave his presentation on what happens in the US, there are 3 projects with SENS, Noted, and the ESNet High Touch. One question which was in my mind is, what about the international aspect of that? And what about the international collaboration? So I hope that we will soon hear how we really collaborate on an international level to use the global research platform. Thank you.
Thank you, Asif. Yes, does anybody question about that? Okay. Yves, are you okay?
Yeah, perfect. Thank you.
Yes. Anyway, thank you. And we're going to the next presenter. Okay, see you and talk tomorrow.
Thank you.
Thank you. And I want to explain, I want to introduce the next presenters is Nora Silla, Wati Abdul Hamid from the UPM in Malaysia. And she is one of my the co-applicant to the Asia Connect project, and we set up the research platform by Asia Connect project in UPM, and she explained about the UPM strategy and the future works. Asila, are you there?
Yes, I'm ready. Can you hear me?
Yes, so we can hear and watch.
Okay, thank you, Jane.
Yes, go ahead.
I need to share my screen, but I cannot share my screen.
I have to.
Yeah, thank you, Yves.
It's Lily's your presentation.
Okay, you're there.
Okay, thank you, Yves.
Awesome.
Okay, so I believe everyone can see my screen right now.
Yes, that's perfect.
Okay. Wait. Yes. Okay. Thank you, Jung Hoon. My name is Nur Asila. I am from University Putra, Malaysia. So as what Jung Hoon said just now, we are one of the partner in the high bandwidth distributed HPC. And as what you've mentioned just now, Malaysia is one of the countries that benefited from this project, especially for University Putra Malaysia. So for today, I'm going to talk about, I'm going to recap a bit from what I have presented for the APAN56, just to see what is our progress. And then I'm going to share with all of you about the Malaysia current, status on these HPC and AI. Okay, so this is the project on the building high bandwidth distributed HPC. So during that time, we discussed all the activities, and then the most highlighted is actually when we're having the conference, that we held the conference in June, and we invited many researchers, particularly from University Putra Malaysia, from various fields, by informatics, engineering. So there are very high demand on these HPC resources. And so we are glad we are in this project. So during that time, the problems, during that time and until now, we are still having very scarce resource on the HPC, very limited on the HPC resource. So most of the researcher who are in need for the HPC, so how they, their pattern or how they fulfill their need is either they go for cloud, or they just bought resources that just enough for their research. So it's quite wasted in terms of not sharing the budget, and so we can get more resources. So during that APAN, I also highlighted that we're going to do a trial run on the building distributed HPC platform in September. And we're glad the system is completed, and right now it's under trial progress. We did some trial with several projects, and we also highlighted that we're going to organize focus workshop based on focus disciplines. And we are also glad that we're going to have this first workshop next week on 8 February, Thursday. And for the awareness for the HPC beginners, because we need to create this culture, especially for the non-computer science background, is always in progress, and is going to include it on the HPC series workshop. Okay, so this is actually, I just want to highlight that what is our progress based from our last APAN56. So done recap for the APAN. So right now I would like to share about Malaysia current situation. So recently, I can say about 3 to 4 months ago, Malaysia Prime Minister and the nation is actually being emphasizing about the AI. As you can see here, there are 3 items here. I mentioned about the YTL going to power the AI with NVIDIA, and then our Prime Minister Awan Ibrahim is actually really emphasizing on the AI projects to the academia and also to the citizens. And for University Putra Malaysia, our main focus is actually on food security and agriculture. So I'm going to go detail for each item. So this is one of the news that saying Malaysia is ready for the rise of AI. So in order to perform this, in order to implement the rise of AI, so you can see from this photo that I guess this is happened last December, or I think last December. So the visit from NVIDIA CEO to Malaysia, and you can see this is our Prime Ministers, and with the YTL, so they perform a collaborations to develop the AI data center for Malaysia. So they expected that it going to be operational in mid-2024. And then this is actually the platform. So in Bahasa Malaysia, it mentioned here AI untuk rakyat. So AI untuk rakyat is, in English, means AI for citizens. So this platform is all in Bahasa Malaysia, in Malay. So they provided this platform for our citizens to introduce for the awareness about the AI era, what is AI. So this is how I can say our Prime Minister really emphasize on the AI project for the citizens. So there are several conferences being organized to showcase the AI project from the universities and in the industries. So this is actually focused on UPM, so I mean from my universities. So we've been assigned from the ministry that our focus is more on the food security and also for the agriculture. So University Putra Malaysia, in the beginning, P is actually for pertanian. Pertanian in English means agriculture. So I believe that is the reason why, because we have agriculture faculty, forestry. So I believe this is why we have to focus on these items. And then we have to link everything AI with these two focus. So this is one of the example then being showcased during the conference for AI for Rakyat or AI for citizen recently. So this is just some example that I just want to share. But I can see most of the HBC resources currently are still in development planning, as you can see from the YTL, and even I did call them just to get collaboration, but they are still not really having like a group or committee to perform. So most of the projects are still in developing, and there are several resources from other universities also are still in progress. And I can see that once all those facilities are completed, probably we have to pay. I guess it's highly probability that we have to pay for that facilities. So what I can say that we really glad that we receive or we join. We are one of the partner for the building high bandwidth distributed HPC. So we've managed to get the resources, and the resources is ready. So at least for UPM, University Putra Malaysia, we have the resources to perform all the AI projects that have been assigned from the governments, and also beside the AI. So I don't want that everyone thought that we only have the AI project. So we also have a presenter, the next sessions. Dr. Fiza is also from University Putra Malaysia. So she is actually representing from the bioinformatics. And we have many other projects related to machine learning, and then engineering and also agriculture. So what I can say, you are thankful we are involved in this project. So we expected that we can perform some performance analysis and some report in the APAN57. We hope that we can provide those report with the current progress on the APRP.KR platform. And also we really hope that this project can be expanded to have more nodes in order to support more needs on Malaysia AI projects and also other projects. So I guess that's all from me. Some updates on what we have been doing for the APRP projects, and also from Malaysia current progress. Thank you.
Thank you, Attila. Yes. Does anybody have a question about that? Okay. And thank you again, Attila. Talk more.
You're welcome.
Okay. And the next present is on me. Mm. I'm sorry, the Zoom restart. Mm. Yes, just to Zoom is drop down right before. Why not the restart? Mm. Yes. Mm. Oh, yeah. Mm. Just starting and just waiting signal. Okay. It's okay? Yes, I joined? Okay. Okay. Okay, let me start. The final, the presenters is in the session one is, my name is Jung-Hoon Moon. I came from the KIST in Korea. The KIST is the Korea, the National, the Representative Supercomputing Center, and the Research Network Centers. I belong to the Research Network Center. The name is KoreaNet. Today, I want to talking about is a Korea Research Platform and the future works. This is today's my contents. The first one is I wanted to announce this the background of KoreaNet, and the second thing is introduce of a Korea research platform. It means that it's about the KRP and HPC over HPN or something like that. And the third topic is a related project from the research platform, and finally the conclusion. The background of KoreaNet. This is the domestic, the KoreaNet, the research network maps in Korea. we have a maximum is 1.2 terabps backbone and 17 local gigapops for domestic. And among Seoul, Daejeon, Gwangju, Busan, Daegu, and between Seoul and Daegu and the 400 gigabps connectivities. And we have the 5 gigapops for international as the 100 gigabps, Hong Kong, Seattle, Chicago, Amsterdam, and Singapore. And the Korean is 24 and 24 hours, and the 365 the operating service. And we the remarkable things is we have over 200 members of institute. And this map is the globally the how to connectivity as I talking about before. And the Korea is a small country, but the yellow line is 5 international gigapops based on 100 gigabps. The reason why is why I introduced is the Korean background. The research platform is the meaning is the we need big data superhighway, and at least 100 gigabps connectivity is very important to make the research platform. And the CREONet and the advanced countries research network is already over the 100 gigabps each, the one another connectivities. So the transmission is it seems like it's a local machine to transmission over the continental. And this table, this is the 17 gigapops location and the places, and this picture and the summary of the big data, the applications in Korea based on the research institute. The Korea has 25 the government-funded research institute, and they have their own research facilities or analysis facilities, and they own their research. All those the institutions are connected with the CREONet. So the high energy physics, astronomy, space, and the canon bio and the meteorology, K-STAR IMISTES is the fusion research, nuclear fusion research, and the basic science facility, education, building construction, and the particle accelerator. And all of those applications can be running on the research platform. And from now on, I want to explain about this Korea research platform. And this picture is the finally aim to the KRP, Korea Research Platform. In the middle box is establish the high reliability and the high speed transfer system without boundaries between participant institutes. In the middle of a circle is, as I mentioned before, and the 25 the government funded research institute. Luckily all those institutes are located near our key city. It means that our connectivity is well working and very easy to extend the branch the institution research labs. So this project is since 2015 currently, and small steps to expanded to our neighbor institute. And the final goal, as I mentioned, is no limitation without boundaries. Because Korea is, as you know, the separate from North and South Korea. The Korea government is big pressure to the security issues to all of the nations. So even though the research institute, for example, for chemistry, they have 100 gigabps research network lines, but it's someone's outside is the access to inside their institute. They cross the 7 security check barriers. It means that is the very restriction, the high capacity, high throughput performance network. Even though they have 100 gigabps network, but their performance is around 20 or around the 30 gigabps. It is a very unfamiliar situations. So we wanted to without the boundaries between participant institutions. So this picture is the technical aspect. The Korea research platforms look at the inside and the deeply. It means that is HPC over HPN, the high performance net computing over high performance networking. As I mentioned before, the KIST is the Korea Representative Supercomputing Centers. Is in the middle of the system, REON is the Korea, the sixth is representative supercomputer. We have also the GPU machines, and we have also the storage center. And we want all those the research facilities, computation, analysis, and archiving, and storaging, and to share. It can be shared through the research platform. So this is the basic architecture of the Korea research platform. The right side is distributed computing, and all those machines is managed by this Kubernetes. The Kubernetes is the strongly the management, the open source, and the computing part is the transfer part, is monitoring part, and the logging part, and the storage part. All those consist of this the Kubernetes worker node, and it means that is all this platform working based on this container technology. And the right side is the what kind of the open sources involved with this platform, and how to establish the web UI and the login system. And this is the recently our the accomplishment for the third-party areas is a big data-based API network through the research platform. And the workflow based on the research platform, it means that in the separate user and the management line, and the combination of the machine learning and the AI and the ensemble relations. So many the workflows is the complete with the user line and the management line. But this is the support for this ensemble model. It means that is from A model's output is the next B model's input, and we make this workflow and through this the 7 APIs. The API communication in the container, and it's very the nature, the flow is to make the workflow. There's the one the model's input or output, and it can be using the next model's input or output. This is the testing of the ensemble model. There's work node 1 and work node 2. There's all those work nodes is controlled by this Kubernetes, and the adaptations is the 7 the internal the communication API technology, and all the data and the workflows is set up these those protocols. And this is a table of the workflows. And this is the actual coding from the Python. And so far, I just explained about this. The Korea research platforms is the core, the body, and the recently the application areas. It means that the API, the network communication, and from now I want to explain about the related project. Our Korea research platform is involved or the support for third-party areas, the project or contribution. And the first thing is Asia Connect project is it just 1.5 years. It just finished the last year, and involved is 4 countries: is Malaysia, Australia, Korea, and Pakistan. And Australia, Korea is the advanced country as advanced countries to share our experience and donation the computing resource. And the Pakistan and Malaysia is underdeveloping countries, and we can the helping or the support the computing resource or the AI resource and our experience. The reason why this project is main goal is the so many countries in ICRs, they have the very smart third-party researchers, but they don't have enough computing environment. IT infrastructure is the really not enough to support all scientists this desire. So this meaning is the the contribution to the small servers each country is together the 1 point to make through this Kubernetes and using the big data superhighway, and the third party researchers using this platform as a single machine. The Ivo Pops is from this before the presentation. He announced the NLP or PRP or TNLP, something like that. And in case of the US, the NSF and the San Diego Supercomputing Centers assumed that all countries of the US it means that is 50 states that involve the US the research the platform. So we want the same motivation, the NLP or PRP, and we want to set up the same research platform in Asia areas. And this is the overall pictures. And the user case 1, 2 is AI science and the bioinformatics, and USA, Korea, Australia, and the donation and the sharing experience and providing HPC through the high performance network, and using HPC parties Malaysia and Pakistan running their jobs on this platform. And we have the several the 3 activities. So first activity was building a big data superhighway based on TAIN. And activity 2 is building a distributed HPC platform. Activity 3 is presenting pilot user cases. And this is the activity 1, this picture, overall picture. And this is activity 2, overall picture. In the middle of the box is the high bandwidth distributed HPC platform. And the third pictures is the application areas. And the related project 2 is smart agriculture. And the Korea Rural Development and the administrations wanted to deploy this system and this research platform to their areas. The title is Development of Integrated Linkage System for Agricultural Big Data and the Utilization Model. The key part is the agriculture big data transfer and the running cloud model on the research platform using CPU or GPU. And the establishment is a research platform for end user. It means that the farmer and the developers, it means the agriculture researchers. And the related project, this is the architecture is of the core, the platform inside. It's almost similar, this KRP platform. And this is the real, the server's location. And the right rectangle means that is the logically separated and distributed. And this is interface. This is the this set models is cram models, how to running, and what about is the result. And this is the tomato, the growth prediction. And the related project 3 is a smart decision-making system. It comes from the Korea Environment Institute project we granted 2022. And the title is Establish an Integrated Impact Assessment Platform and building an ensemble of multi-climate scenarios based on the new climate regime. It means that's AR6. The KIST, the support parties, building a container-based research platform and the self-storage. All the below is almost the same as the agriculture areas. The Environment Institute or the Environment department want to do the smart decision. They want to consider the climate and the earthquake and the population and some other aspect. The gathering the one machines that they want a learning model, and they want making very smart decision to the public, but they don't have the proper system or a proper computer. So this project is the many part is joined. For example, is computer science and the climate researchers and agriculture researchers and environmental researchers. And this project is very meaningful for in the future in Korea. And the related project 4 is a smart hospital. It's almost the same, the conceptual. The title is Construction of a Patient-Tailored, Hyper-Personalized Digital Medical Ecosystem based on Omniverse platform. And it means that it's the remote patient and the doctors meet is on virtual spaces, and they want to look at the patients is the private data, and they want a smart decision. And the KISTI, the support area is a big data superhighway for inter-hospital and the human and genome data centers. And then, I want to briefly showing to this the Korean giant group researchers. So far, there's the 4 related project is made, but this research group is the as I mentioned the early staging, and at the table the big data research areas. So this is astronomy areas, is how many the institute and what kind of activities running on our Korean. And this is HEP, high energy physics areas, and this is weather and climate areas, and this is genome and bio areas, and this is K-star areas, and this is electronic telescope areas. So so far this I wanted to explain about this Korea research platform, how to build, how to apply this, what kind of users want to use in the research platforms. And then there's our conclusion and the future plans is just one thing is the expanding to our neighbors and our the institute, such like as I mentioned before, is 25 Korean national institutes already exist. They always not enough the computing resource and GPU resource, and how to handle all of these infrastructures. But the KISTI is IT areas we can support for third-party research application areas. The KRP is will be going to the next stage with the application areas. Thank you for your listening. Anybody question about that? Okay, and its time is 10 minutes over. Sorry for that. And our session 2 is started 3:30, and the session 2 include the wireless communication as the application, and the quantum safe network, and the supercomputing centers, and something like that. As anyway, as more people and come again here. Thank you. We take a break.